Hello everyone, and welcome to our online lecture series in plant biology. Today, we begin upon the first true material in the class, going over some of the topics that will build the foundation for your understanding of plant biology and botany. This course is a broad survey of everything plant-related, and is therefore divided into two distinct chunks. The first half of the class will explore all of the cellular components of plant biology. What is a plant cell? How do they differ across the plant body? How do they grow? What do they do? And how do they make a plant unique from all of the other different life forms on planet Earth, with a special emphasis on what makes plants different from animals. The second half of the course will focus on plant diversity, understanding how plants take this basic building block of a plant cell and use it in different ways, specialize different tissues to create the broad and incredible biodiversity of plant life that we see on Earth today and in the fossil record of the past. So, without further ado, let's get to the material that you will be expected to master in today's plant cell lecture. So we could not begin talking about plant cells without a broad introduction to the idea of cell theory. Because it is from this observation that all life is made of cells, that we began to better understand the building blocks, the functions, and therefore the ability to manipulate plant life on planet Earth today. So whether you are going into education, agriculture, horticulture, plant breeding, genetics, biotechnology, all of these fields will necessitate a good understanding of basic cell theory. Cell theory began with the invention of the microscope. An individual by the name of Robert Hooke invented the microscope. While this lecture is online, I would advise that you do take your own notes as you're going, so you'll see me writing things down. And I'd advise that you not only write what I do, but also go beyond to copy some of the spoken word that I am saying, but not exactly writing. You'll notice that it is very difficult for me to both speak and write at the same time, so I will take brief pauses to emphasize material in my writing, but you should also be going above and beyond and copying down some of the further details, so that way you can tell me the story that I gave in lecture. That's right, I do expect to some degree that you are able to recapitulate or retell the stories, the histories, the facts that you are getting in lecture in order to succeed on the many different forms of assessment in this class. So, back to our story. Robert Hooke was an English scientist in the mid-1600s to early 1700s who was responsible for the invention of the microscope. Now, he did more than just the microscope. If you've taken physics, you might know Robert Hooke from Hooke's law regarding springs. But in terms of biology, his major contribution was both the invention of the microscope and coining the term cells. So here we have an image from your textbook showing some of the early models of Robert Hooke's microscope. You can see that this is a very primitive device compared to what we're using today. Just a simple magnifying lens. But you can also see some of the early images that Hooke may have seen, or like what Hooke may have seen underneath his early microscope, showing two different plant tissues. Now, from his observations under the microscope, he observed that all plants are made of tiny little squares, and he called these squares cells. Now, it would be a while before Hooke's tool of the microscope was applied to a wide variety of different organisms. After Hooke, and about 100 years later, two scientists by the name of Schleiden and Schwann, that's Schwann. And if you have any question on the spelling of words, note that I do understand that my handwriting does leave something to be desired, but you always have typed out vocabulary sheets that you can access in order to make sure that you are spelling things appropriately, as I do think that spelling is an important component of any foreign language, and to some degree, biology as a field should be studied as a discipline like a foreign language that includes topical applications. So, Schleiden, make sure we're spelling that correctly as well. Schleiden and Schwann were the two scientists who took the microscope and showed that both plants and animals were both made of cells. So not only did Hooke's observations apply to a single sample or the couple samples that he looked at, but they did a very systematic study of tons of different animals and plants and saw that all of these organisms fundamentally were made of these smaller building blocks. It would be a while later for a third scientist, Virchow, spelled Virchow, to make the observation that it was cells that made other cells. Today, in a process that we call mitosis. So in these three major observations, we can synthesize cell theory. Biological cell theory, and therefore the cell theory that applies to plants, states that first and foremost, life is made of cells. That these cells are the basic building blocks of life. The second point of cell theory states that life occurs within cells. So the fundamental processes of things like metabolism, breaking down and building the macromolecules necessary to perpetuate life, eating, drinking, all of that is going to occur within a cell because the cell is the basic building block of life. Third, addresses the origin of cells. Using Virchow's observations, the third point of cell theory states that cells make cells. That cells arise from the duplication of other cells. And as an addendum for this third point, either 3.5 or a new point 4, that cells have the hereditary information necessary to make all future life. And so this cell theory began by observations underneath a microscope by Robert Hooke and built upon by his colleagues, even if they were far after his own lifespan, really put into place our understanding of what it is to be alive and what we need to understand in order to truly get a good understanding of what makes a plant. Because a plant is built up of these tiny cellular building blocks, we need to start our discussion of plant biology by looking at the plant cell. So before we get into that vocabulary, I have a few other terms that I'll be throwing around. And again, if you are questioning or don't know any of these particular vocabulary terms, I'd encourage you to pause the lecture. You can utilize both your textbook, as images like this are found in your textbook, or Wikipedia. Those are both excellent sources to do further reading on topics. I will note that the textbook at times can be fairly dense, so I would encourage you, at the very least, to read the end of chapter summaries. Those are 1 to 2 page summaries of everything discussed in the chapter at a very introductory level of understanding. So hopefully that encourages you to engage with your textbook this semester. Pretty please do so. It will help your grade, I promise. It will help your grade and your understanding of this material. And I hope that you are taking this class not only for the grade, but for a better understanding of what builds plants up. So our next two terms regard the different types of cells that we will encounter in life and to some degree in this class. Those are our prokaryotes versus our eukaryotes. To understand what these terms mean, we have to dig into our Greek roots. The root pro means before. And the root karyo or karyon is kernel. Whereas in eukaryote, EU is true or good. And then again, karyon is kernel. And what we're referring to in before kernel, or the true kernel, is the presence of the nucleus. Eukaryotes have a true membrane-bound nucleus. Prokaryotes have no nucleus and no membrane-bound organelles. And so practically, what this means, why does it matter to have a nucleus or membrane-bound organelles, is that each of these membranes allow for separation, for specialization. When you have something bound in a membrane, the functions that occur within that membrane or within that bag can be different from the functions that occur within the main juice of the cell. The juice of the cell we refer to as the cytoplasm or the cytosol. Your textbook kind of uses them interchangeably. But rather than having one major soup that all of our metabolic reactions are taking place in, like in prokaryotes, eukaryotes can have cell specialization, where each different membrane-bound organelle can do something slightly different. So you will generally see prokaryotes as much more simple and eukaryotes as much more complex. In the same way that you, with all of your internal organs, are able to do different functions in your stomach versus your kidneys, versus your intestines versus your brain, whereas which is akin to a eukaryote, whereas a prokaryote would have all of those functions of thinking, digesting, and filtering done within one bag, and therefore is far less efficient or capable. Organs and subfunctionalization allow us to be more complex and more specialized. Now, some of the other features that divide our prokaryotes and our eukaryotes are the structure of our DNA. Going back to cell theory, we see that the hereditary information is our DNA, or deoxyribonucleic acid. Deoxyribo refers to a sugar backbone, and nucleic acid is the particular form of information storage. So from previous biology classes, you may be familiar with DNA as the long-term storage, or RNA as the short-term temporary storage of hereditary information. Eukaryotes have linear DNA that you may have seen drawn as chromosomes in the shape of an X. This means that they have a start point and an end point. Contrast this with the genome of prokaryotes. They instead have circular rather than linear DNA. There's a lot of cool applications that we'll get to later on in this class, like genetic modifications, where circular DNA plays a big role in this. But for now, I just want to introduce y'all to the fact that prokaryotes have circular DNA and eukaryotes have linear DNA, packed as large chromosomes. We also, in those linear DNA strands of eukaryotes, they are organized with histones. These are packing proteins for our massive chromosomes. To keep things organized and from getting tangled, you can think of that linear DNA as a set of headphones in your pocket. If you throw a set of headphones directly into your pocket and they go about living your life, they get very, very tangled. That's not good for DNA. Histones allow for that headphones or DNA to be placed in your pocket and organized throughout your daily life. So histones are something that enable our eukaryotes to have those long, large, linear chromosomes. Generally speaking, you will see prokaryotes as very small in size, whereas eukaryotes are much larger in size. There's something like a 10 to 100 times size difference between prokaryotes and eukaryotes, just to give you an idea of the scale of the difference here. Our last compare and contrast, or really contrast, feature of prokaryotes and eukaryotes is in a very particular organelle. That organelle is a ribosome. Ribosomes are our protein factories. They make all of the proteins in our cells and in plant cells and in bacterial cells. Proteins are the machinery, the tools that you need in order to live. So ribosomes are very, very important. And in order to help us distinguish between prokaryotes and eukaryotes, we can use a structural feature that separates the ribosomes of these two major different groups. In prokaryotes, you have ribosomes that are 70S in size, whereas in eukaryotes, you have ribosomes that are 80S in size. Now, I won't just yet emphasize what exactly that means, or just know that this is a size difference. You can think of S as a unit, kind of like centimeters. But prokaryotic ribosomes are smaller, and eukaryotic ribosomes are larger. Hold on to that fact up until we get to our second half of the class. That's where it'll play a much bigger role. So, playing the long game with that one. So here are our major differences between our prokaryotes and eukaryotes. We see that prokaryotes are, I'll just shrink this down to keep this to one page. Again, you writing this out on pen and paper hopefully have the ability to add space in your notes. You should additionally, I would strongly recommend that after listening to one of these lectures, maybe a day afterward, try to resynthesize your notes by writing them out again with less detail. Each time you rewrite your notes, you solidify the information and set yourself up to better succeed in this class. So, going back through prokaryotes versus eukaryotes, we have prokaryotes being the before kernel or no nucleus group. They are generally much more simple, smaller in size, have circular DNA, and smaller ribosomes. Eukaryotes are much more complex generally. They have a true kernel or true nucleus. They have linear DNA organized in chromosomes, with histones helping wrap that DNA. They have much larger size in general. These cells that Hooke was looking at were all eukaryotic cells, much, much bigger. And their ribosomes are bigger as well, with the size of 80S. Whew, okay. So that's the two different types of cells that you encounter in this class, either prokaryotic or eukaryotic. So let's look at the different classes of cells and what the organelles of cells are. And organelles, while I do not know the true origin of the word, I do know that organelles were, the term itself was coined by German biologists who were first observing some of these smaller cellular features. And in some certain German dialects, they have a tendency to add "elle" to the end of the word to make it cute, in the same way that we in English use the ending "y" to make things cute, like pup to puppy. They add "elle" to make the term cutesy or smaller. And so it is my, at least, devotion to my memory here, to help me keep track of this, that an organelle is an organ, but smaller, and in many cases, cuter. So now we'll be going over the different types of, different classes of cells, and the organelles that are shared by both of them. Our two classes, of course, being plants, which will be the focus of this class, and animals. So kind of give you a compare and contrast of what things you see in one versus what you do not see in the other. And again, I will, for this next part, assume that you have some background in this topic. So if you don't, please make sure that you go back and try to build on some of this vocabulary. Your textbook, Wikipedia, can help you do so. But let's get through these different organelles. So both plants and animals have the equivalent of a cell brain called the nucleus. The nucleus holds all of the DNA and therefore is oftentimes considered the brain of the cell. Both have a nucleus that has a nuclear envelope, a membrane that helps protect the inner contents of the nucleus while also allowing for quick transit. The nuclear envelope is defined by having nuclear pores, these constantly open surfaces that allow for quick movement of very particular things in and out of the nucleus. Both plants and animals have an outer bag, something to keep their insides separate from the outside world. That bag is called a plasma membrane. It is this plasma membrane that will determine what goes into the cell and what stays outside. Both plants and animals have the ability to move things inside of their cells via a cytoskeleton. The root of this word is that it is the skeleton of this cytoplasm. And cytoplasm is a term we've already introduced, cytoplasm being the cell juice. So we have the ability to move things around in our cytoplasm because there is a skeleton present in there. Both plants and animals also have small compartments, small bags to move things around. We call those bags vesicles. And these bags are made in something called the endomembrane system. The endomembrane system is the combination of two major organelles, the endoplasmic reticulum, or forevermore, the ER, and the Golgi body. These two structures will go about like a UPS store producing or a UPS store plus Amazon warehouse, producing, packaging, and getting things into vesicles, and then the vesicles will get them where they need to go. So when I talk about the membrane specialization, I'm really referring to a lot of what's happening in these vesicles, endoplasmic reticulum, and Golgi body of the cells. Now, in terms of what differentiates our plants from our animals have a organizing feature called a centriole. This centriole allows for animal cells to split in the process of mitosis. You can think of a centriole as a conductor, because an animal cell is generally lacking any particular structure. They have a soft outside, therefore you can think of animal cells more as a blob in shape. They need a conductor in order to dictate where things move in the animal cell splitting. Plant cells instead have a cell wall. Now you may know a plant cell wall as the outer protection of a plant cell, and you'd be right. But because they have this firm outside, they also have a better idea of how to split their cell. They adopt oftentimes a more set, frequently rectangular shape, or at least that's what the cartoons of plant cells would have you believe. But the plant cell wall acts as a helper in mitosis, as well as giving the plant a lot of defense and structure. We'll go more into the exact building blocks of a cell wall after we get done listing all of these different organelles. And no small portion of this class will be spent talking about the plant cell wall, because so much about what makes plants unique is held within that cell wall. Plants also have a vacuole, or a giant bag of water to help regulate the amount of water versus the amount of salt inside the plant. Pretty important to have that as a regulatory element if you don't have a bloodstream to regulate all of your water. Plant cells also have chloroplasts. The separate bags that we're going to spend the next several lectures discussing, because this is our bag of photosynthesis. The unique process by which plants breathe in carbon dioxide and produce sugar. And plants also have a peroxisome, whose function we will discuss more because it's tied in with the chloroplast. The other feature that I oftentimes see lumped in to animals and excluded from plants is a mitochondria. Now, hopefully you had a knee-jerk reaction that the mitochondria is the powerhouse of the cell. And we oftentimes assume that plants do not have mitochondria. But I left that off because I want to address this point head on. As a plant biologist in this course, you are to never forget that plants also have mitochondria. They too need to generate energy, and they do so with the help of the mitochondria, the bag that performs cellular respiration. A concept that we won't go into in this class, but I might encourage you, if you are interested in it, look up some YouTube videos in it, or take one of my other biology classes here at West Shore Community College, where we do spend different amount of hours, anywhere from 2 to 6 hours, discussing the details of cellular respiration. It's a fantastic process that warrants a fair bit of extra studying. It's pretty cool, I think. So, those are our different organelles. Both plants and animals have mitochondria, nucleuses that have a nuclear envelope, plasma membranes that keep things in or out, a cytoskeleton that moves things around in the cytoplasm, and then the different components of the endomembrane system: vesicles, endoplasmic reticulum, and Golgi bodies. Plants additionally have a cell wall, a vacuole for water regulation, chloroplasts that perform photosynthesis, and peroxisomes that help with some function of photosynthesis. Now let's take a look at some cool drawings of what actually makes the plant cell wall on a molecular level. You'll notice that every single topic in this class we keep getting more and more detailed on a building block level, because we need to understand how these things function on their smallest level to see how they work on their bigger levels. So a plant cell wall can be illustrated basically by my little cartoon here. You have a couple different features. In black, you have what is called the primary cell wall. I'll abbreviate cell wall as CW here. This is the initial building block. You can think of it as a single layer brick. You have this illustration being two different cells. So cell number 1 here on the right and cell number 2 here on the left. And the black is the primary cell wall laid in between those cells by each of those cells. Single layer allows for separation, but not particularly strong. In order to strengthen that separation, you have, in gray, very thick secondary cell walls. These secondary cell walls are kind of impenetrable. There's no way to pass in between them without a ton of effort. We have gaps in those cell walls that we call our pit cavities. And in those pit cavities, we have a very unique plant feature that will be our last organelle listed on this plant comparison sheet. That organelle is a plasmodesmata. And we know these as our plant cell-to-cell connections. And the best way that I can help illustrate what makes a plasmodesmata in those pit cavities different from what we see in animal cells is through kind of a gruesome description. So animal cells are held together by structures that act like holding hands. We have proteins on the outside of our cells that hold on to proteins from other cells. So animal cells just hold hands and hold on to each other. Plant cells are what you might consider a little bit more committed. Rather than just having proteins that connect to one another, plasmodesmata are the equivalent of veins running from one cell to the other. So you can imagine this, instead of you and I holding hands to be connected, us holding hands, and additionally, my blood vessels running into your hand and your blood vessels running into my hand, so that we have a blood-to-blood connection between our plant cells. While gruesome, it does help to illustrate the level of connectivity that we get between our plant cells. And because there is this incredible connection between plant cells, it has huge impacts on downstream things like disease transmission once you get into a plant. So I introduce you to plasmodesmata here and now, so that way you have a better understanding of how disease transmission works later on. The last component of the plant cell that you see illustrated here is the middle layer, or in Latin, the middle lamella, a structure in between the primary cell wall that allows two cells to be connected. Now, as a structural component of this cell wall, we have the different sugar fibers that make up this cell wall. So when I draw that black line of the plant cell wall here, I want you to know that this is the structure that we're actually looking at. The middle lamella is on the outer layer, and it's a soft connection between our cells. On the inside of that primary cell wall, we have pectin, which those of you who make jam may know pectin as a gluey substance. So it is a softer but gluier substance that connects our cell wall. And then two major fibers, cellulose and hemicellulose. And I want to take a moment here to highlight cellulose, to highlight again, to highlight it a third time. It is a term that you need to be familiar with. It is the primary building block of cell walls. It is a sugar molecule. It is the thing that gives strength to cell walls. And I will use it a lot in this class. So let's go ahead and highlight it a fourth time. And please do keep track of your cellulose cell walls. Cellulose and hemicellulose are two different sugar structures that provide strength to our primary cell wall. And then beneath a primary cell wall, you have that plasma membrane, that living structure that's giving directions as to what stuff is going in and what stuff is going out of the cell. Now, in terms of your secondary cell wall, we're going to be using two terms, two sugars, very frequently, or three sugars, sorry. Those sugars in our secondary cell wall are lignin, cutin, and suberin. And each of these have different degrees of water permeability, or blocking of water. Suberin is cork, literally. So those of you who have ever popped a bottle of wine know that the cork in the top prevents any water from escaping. Suberin is very, very good at preventing water from leaving cells. Cutin is a waxy substance on the outside of plant leaves to help prevent loss of water. And lignin is a very strong substance that also helps prevent water loss and allows for structure within the plant cells. So we'll be revisiting lignin as it is important both in helping plants move water around and helping them stand tall. We would not have trees without lignin. In fact, all plants would be about the height of a moss if it were not for this secondary cell wall substance of lignin. We have this additional drawing from your textbook showing middle lamellae as the layer in between, the secondary cell wall as very thick structures, the primary cell wall as the primary outline, and then each of these individual cells. You have the same zoomed in feature that I drew over here. So whether you like the book cartoons or my cartoons, I do hope that you have a better understanding of the organelles within a plant and animal cell, what differentiates them, and how the cell wall looks. So now, as a bit of review, let's go ahead and label these different features in a plant cell from your textbook. So from going clockwise around this image, we have our first gaps in the plant cell wall here. These are our pit pairs or pit fields with our plasma desmata. You have pancake-like stacks that are part of the endomembrane system. These are our Golgi body. You have the bag that holds all of our DNA, our nucleus. You have the bag that's performing cellular respiration, the mitochondrion. You have the bags outside of the nucleus that are part of the endomembrane system. The ribosomes that are on the endoplasmic reticulum. So ribosomes plus our ER. This blank right here is a lot of extra terminology, but we can go ahead and see that it is our endoplasmic reticulum here. I won't ask you to know the difference between smooth and rough. That's for a different biology class. Continuing on, we have our structure here on the very inside layer, the living layer of the plant cell that tells things what comes in and what comes out, our plasma membrane. We have the cell juice, or cytosol, or cytoplasm. And then we have tons and tons of green bags, lots of them throughout the plant cell. These being our chloroplasts. We also have the middle layer in between our two primary cell walls, the middle lamella, and our two cell walls that differentiate them. And then we have our giant bag of water, the vacuole, whose membrane gets a specific name, the tonoplast. Now, I won't fault you if you call it a tonoplast or a vacuole. Two names for kind of the same thing. The vacuole is technically the bag. The tonoplast is the plasticky part of the bag. There is a distinction, but far too granular for what I expect you to keep track of in this class. And then we have our last membrane-bound bag that's associated with the chloroplasts, our peroxisomes. Whew! So kind of a quick bird's eye view of a cell drawing, but you have additional ones that you could go online, and I hope that you can identify these different cellular features, whether they be on this cell drawing or any other. Now, our last bit of terminology today, our last three vocab words are our three plant tissues. We'll be talking about them a lot because our plant cells specialize into plant tissues in different ways. So those three plant tissues are the epidermis, or your outer layer, the skin of the plant, made to help keep all of the water inside, the ground tissue, the living layer. This is where a lot of the metabolism is happening. And metabolism just being all the cellular functions you need in order to live. And then the vascular tissue layer. This is your transport. So in your vascular tissue, you're going to be moving around sugar and water to help keep the plant alive. It's kind of like your blood vessels, but vascular tissue has directionality. Your blood vessels go in a large circle. So those are the three plant tissues: the epidermis, the ground, and the vascular. Our very last vocab term for today is parenchyma. We will be discussing it in more detail later, but chyma refers to your liquids, your living substances. So the Greeks used to think that we were made of, our chyme was our blood, water, everything mixture, and you needed to make sure the chyme was balanced in order to stay alive, and you died when chymes were unbalanced. Thank goodness we've come a long way in terms of our understanding of the human body, but the suffix of the chyma still applies to the different type of ground tissue that we see in plants. And parenchyma is our living ground tissue, that performs photosynthesis. And so our next 2 lectures will focus on what's going on inside of that parenchyma. Because from here, we take on probably one of our most molecularly challenging concepts in the class. I like to get this out of the way early while everyone is nice and fresh. We're going to be talking about photosynthesis, of course. So, hopefully you've enjoyed this introduction. Again, if there's a lot of terminology that you do not know, please reference your textbook, Wikipedia, ask me questions, ask your classmates questions, build up this vocabulary so that way we can all start at the same starting point. I do not expect you to have a strong understanding of biology before entering this class. But after this first lecture, I do expect you to be familiar with these terms, because this is the platform that we're all going to be standing upon to go forward. I'd encourage you to look up the different summary tables in your plant biology textbook, as it gives you probably more words than you need to know. And if there are words that you seem a little bit overzealous, like glycoproteins, let those slide. But see what else you're able to recognize, like cellulose, hemicellulose, pectin, lignin, cutin, suberin, as in terms of other structural components. Reading, talking, writing your notes again. These are the ways to succeed in this class. Have fun going over your vocabulary. I look forward to discussing some of the terminology with you, and I will see y'all next time for our discussion of photosynthesis.